All right, everyone. I think I see some new people today with some familiar faces too. Before we start today, I'm just going to introduce a little bit about the Hmong Association for Scientific Research, and then we'll go to the talk that's scheduled for today. For those of you who are new, who may not know what we are as an organization, so the Hmong Association for Scientific Research, or HASR, as we call it, is an organization that we created as some of the first Hmong people to get PhDs in the sciences in STEM, especially biomedical sciences. And we want to create this organization to hopefully allow us to share some of our work and also to serve as peer support for each other in our careers. And so we welcome a lot of you, those of you who join us today. Thank you for coming. And if you are interested in joining also our organization, please feel free to let us know, and we could add you also as a member. Okay, so that's how we created a couple years ago, and we have about maybe 30 something members now of various levels, from faculty to postdoc, graduate students, and even undergrad. And I think we might have some high school students as well, maybe. And so that's us. And my name is Tu Yevu, and I'll give it now to Pajal to introduce the talk for today.
Thanks, Tuya. Good to see some familiar faces, or not faces yet, but just some names. Encourage you to turn on your video if you feel comfortable. Always nice to see faces to names, but of course, do what you can. I'm so pleased to welcome Dr. Pangkong Fox today. She'll be sharing a talk about her work and her journey to STEM. And so just a little quick brief bio. I won't go into too much detail since I'm sure she'll provide a lot more detail in her talk. Biology, and also was an adjunct professor at Northern Illinois University in Ishwaukee College. And after her youngest son was diagnosed with a rare genetic neurodevelopment disease, she joined the leadership team at the CACNA1A Foundation. And so she's going to go into much more detail about that, and I'm super excited to hand it over to Ping Kong. Please take it away.
Thank you so much for that introduction. So yes, everyone, I'm Peng Kong, or we say it in Hmong Bakong. And I just want to thank Hassar for inviting me. I have not been as active, and I definitely want to try to do that. And I think one of the best ways was to say, "Hey, do you want to give a talk?" So really happy to do that, and talk through my journey to where I am today. So I am the Science Engagement Director for the CACNA1A Foundation. This is a parent-led 501c3 nonprofit that supports those living with CACNA1A related disorders. And this is a rare genetic neurological disorder that my youngest son, was diagnosed with almost, or a little over 4 years ago when he was 6 years old. And so that's really what brought me into the rare disease space. And I think this rare disease space is something that I think all of us kind of realize in the back of our minds, that there are different diseases out there, and there is there are rare diseases. But I think I was trained as an academic, that doesn't really come to mind. And I think I think now looking back, I think, oh, I wish I would have known about rare diseases and maybe invested a little bit more time in rare disease research. So hopefully today I can kind of give you some insight into what that rare disease space looks like, and what I do in my job, and how it can impact different families, and including my own. I'm not going to spend too much time going through my official journey to get to where I was. As introduction said, um, it's pretty normal. After high school, I went to college, and then after college, I went to graduate school, and after graduate school, I did a postdoc, and then after that, it kind of got a little different because I got married at that point, so we were married already when I was teaching, but my husband got a new job, and it brought us into eastern Wisconsin, and I was also pregnant. And at that time, I wasn't really sure what to do as a career. And I had this opportunity to stay home and raise my kids. So that was back in 2014. And it was a hard decision because, I had worked all these years. I got my PhD and I was kind of worrying, what am I going to do? My parents were so happy. They're like, oh, you're a doctor. And I was like, well, I'm not an MD, right? But I am still a doctor. So it was a hard decision. And I had two kids. This is Alex. This is Gabriel. This is when they're much younger. They are 10 and almost 12 now. But ultimately, staying home and being given that opportunity to spend time with my kids was the right thing to do because when my youngest son was born, there were
Pangkong,
some of these. Oh,
Pangkong, sorry are you sharing your?
oh, I'm so sorry.
Yeah,
I guess
I
I'm
didn't
not.
realize I didn't realize you were. Yeah,
I just
please.
jumped right into it here. I'm so sorry. Let's see here. Okay. So you don't really miss much besides my intro slide, but this is a picture of my kids now. Actually, this is from when they were younger, but this is my son Alex and my older son Gabriel. Um, and again, I had the opportunity to stay home with them, and ultimately that was the best thing to happen because my youngest son started showing some delays in developmental milestones kind of early on. He actually had fewer movements in utero when I was pregnant than with my older son. And even as a baby, he was not very mobile. And with my older son, he hit all of his developmental milestones so fast. And so everyone said, oh, this little brother is going to do the same thing. But he actually developed at a very different trajectory. So again, not as mobile as a baby. He was able to sit up without assistance, at 6 to 7 months, but he fell over a lot. He would sort of like look over his shoulder and he'd fall right over. He never learned to crawl on all fours. He could army crawl. He could kind of find his way around, but he wasn't doing it sort of as like a neurotypical kid would do. Um, he was trying to stand up between 1 and 1.5 years. He actually couldn't take any sort of like independent steps till he was closer to 2.5. And I would say he probably officially was able to walk when he turned 3. And so some of the diagnoses that we received for him is that he had hypotonia, which is low muscle tone. He had torticollis, which is this dystonic movement where his head was tilted all the time. He had some eye movement disorders. He had a condition called ataxia, which is a movement disorder, and that's where there's your brain isn't really wired to figure out which muscles to isolate to move, and so it just kind of moves everything. So it's very tiring. In some ways, my son was sort of drunk when he walked. He swayed. He just couldn't find his balance. And eventually, he also had delays in speech, impairments in speech, impaired fine and gross motor skills, cognitive impairment. Once he got to school, he needed more processing time. So all of these things just kind of started building up. And he wasn't diagnosed till he was 6. And so a lot of this was happening before that diagnosis. Um, and so I'll kind of talk through what we call that diagnostic journey that took 6 years in terms of a rare disease diagnosis that's actually not so long. Obviously, we would like to shorten that diagnosis. But in spring of 2017, he was, like 1.5 years old. He wasn't walking, he wasn't talking. And so we were able to get some early intervention through like an early childhood intervention program within the local county. So he received physical therapy, he received speech therapy. He saw an ophthalmologist because he had those eye movement disorders. We saw a developmental specialist, speech pathologist, and so on. And it wasn't until 2020 when his neurologist gave us this tentative diagnosis of left hemiparesis, where your left side is either weakened or paralyzed, and that's under that cerebral palsy umbrella. So in some ways that hit me really hard because I knew what cerebral palsy was and I knew it was permanent. And so I think I think that actually braced me for the actual rare disease diagnosis. So it was a hard pill to swallow, but we kind of felt like, okay, we've got some sort of answer. So we just kept going. We took him to a psychologist. He had neuropsych testing done. And it wasn't until his physiatrist, which is like a physical and medical rehabilitation doctor, she actually said like, hey, I think you guys should get a second opinion because this, he was still able to use his left side just as well as his right side sometimes. So it just didn't feel completely right. And we just kept going through specialists. And then finally, in the fall of 2021, we went to see a neuromuscular neurologist. And this was to rule out any sort of neuromuscular disease because, again, he had that hypotonia, he had that low muscle tone. He wasn't walking on his own, until he was 3. So the good news was this neurologist ruled out neuromuscular diseases, but he also felt that there was some sort of genetic component behind it. So he ordered some genetic testing. Um, and that eventually led to this rare disease diagnosis. So first we had a chromosomal duplication deletion array where they simply looked at almost like a karyotype. You look to see if there are any chromosomes missing or extra numbers or pieces of the chromosomes missing. That came back completely fine. And then we screened a cerebral palsy gene panel. And so this tested, I think, about 256 genes that had been linked to cerebral palsy over the years. And it turned out that my son had a pathogenic or disease-causing mutation or variant in this CACNA1A gene. Less than 200 individuals within the US. And actually, as of today, there are more than 10,000 rare diseases that have been identified. And oh, sorry, I'm missing something here. There should be that 95% of them don't have an FDA-approved treatment. Excuse that typo. So the majority of them have no specific treatments, no cures, and they're sort of just left trying to do trial and error of current medications and things just to treat the symptoms rather than the underlying cause of the disease. So within the US, there are actually, 30 million, actually even more people that are living with some sort of rare disease. And this affects over 300 million people worldwide. So I think there's a fact out there that 1 in 10 people have some sort of rare disease. So it's very likely that you have someone within your friend's network or your cohort that has one or knows somebody who does have one. So again, the fact that it's not so rare is kind of surprising that we don't really learn about those as we're trained, in our academic journeys through the sciences and medical school. So anyway, so we got that diagnosis, and when we asked what to do, we were told to just keep doing what we're doing. So continue the therapies, continue speech, continue OT, and just wait for treatment. So you can imagine as a parent with a young child who was just given this rare disease diagnosis with apparently no treatments and an unknown disease trajectory, it was pretty devastating. But there was what I call a sliver of hope here. The CACNA1A Foundation had just been formed in March of 2020. So, almost 2 years or a year and a half before my son had been diagnosed. And we connected with him. And the amazing thing was that it gave us this community of families whose kids were impacted by this disease. It gave us a community of scientists, researchers who were studying this disease. And so we didn't feel so alone. And I think that's one of the hardest things when you have a rare disease diagnosis is that you kind of feel like there's nobody else in the world who knows what you're going through. And I think there's still stigmatization within the Hmong community in terms of any sort of disease, any sort of disability. And so for my parents, when we told them that, my son had this disease, they were sort of like, oh, I don't think it's real. Like he's going to catch up to his peers at some point. But, we definitely reached a point where it was very very clear that there was going to be this gap between my son and his peers, and it was going to continue to grow over the years. So really happy to have found this community. And then I think the big turning point in my life was that, as a stay-at-home mom, we had this rare disease diagnosis. I wasn't in research, I wasn't in science anymore, I wasn't teaching anymore, and I didn't really know what to do, but I was thinking maybe I could use my skills for something. And I had actually reached out to the foundation about something random, and I ended up connecting with the secretary of the foundation board, and she said, "Oh, hey, you're a parent and you're a scientist." And I was like, "Well, it's been a long time." But they were looking for a science director, somebody to come in and help guide them on the types of research and scientific initiatives that could get us to treatment. Um, so I became their science engagement director, mostly because I didn't have the industry experience, in terms of drug development. Like as academics, we're not trained, to think that way. But I did really love conversing about science. I'm really passionate about what I call conversational science. I think that's something, again, as academics, we're trained to talk at like the highest level about our work, right? But I think we're missing a really big, important gap, and that is how to talk about science to the general public. Because I feel like, especially nowadays, it's like there's this big wall and science is behind it, and the general public doesn't really understand how things like basic science can have improved their lives and can continue to improve scientific understanding of the disease is very empowering. And so that's what I'm trying to share with the families. So the CACNA1A Foundation, again, was founded in March of 2020. So we're coming up on 6 years now. And our mission, I'm not going to read the whole thing here, but really, as you might expect, our mission is to find more specific treatment options or perhaps cures one day for our community. And we really focus and highlight collaboration between different stakeholders. So obviously patients, families, doctors and clinicians, scientists, both in academia and outside, to really find, again, like to make these lives better. And we do that through these three pillars: to advance research, support families, and raise awareness. And so I've been involved in all of these pillars, but as you can imagine, part of my job really is to advance that research. And so I work with our scientific advisory board and our global research network to really think through what our research agenda should be, and how we can execute that. And so again, I said that collaboration is really the key within our mission, and I think this is what drives progress. And it's not unique just to us, right? I think everyone here understands how important collaboration is, and how much more efficient that might be. And so even though we are, a parent-led nonprofit organization or a patient advocacy organization or a PAG, we still interact with many, many different people and institutions. So we do interact, obviously, with those living with this disorder and families, with academic researchers, other patient advocacy groups. We interact with umbrella organizations for rare diseases. So NORD is the National Organization for Rare Diseases. Global Genes is also another big nonprofit that supports rare disease research and also supporting families. We've worked with NIH. We work closely with some grant program directors, especially from NINDS or the National Institutes of Neurological Disease and Strokes. That's the institute within NIH that grant or that funds a lot of the work that our research network does. And we've also had some collaborations with NCATS, which is the National Center for Advancing Translational Research. They also have some rare disease initiatives, so it's been really great to have these resources. And we've also had conversations with the FDA, which is really smart because they're eventually going to be the ones who will either approve or not approve a new treatment for our community. And then we also work with, pharma and biotech companies. Sometimes they already have a drug that might work for our community, so we start talking to them. Sometimes they're looking for a disease in which to invest, and so we kind of try to like sell ourselves and say, hey, this is why you should invest in our disease. And then we also work with CROs or these contract research organizations that sort of do this fee for service. So if we need specific assays done, we can pay them to do those, and that also helps to accelerate their research. Okay, so I'll tell you a little bit about these CACNA1A related disorders. So earlier I alluded to the fact that they are these genetic neurological disorders. Right now, we don't know how many people actually are living with this disease in the US and outside, but there was a publication from a few years that was able to estimate a prevalence, or I'm sorry, an incidence estimation for what we would call de novo or newly arising mutations that aren't inherited. And so the incidence rate of that, meaning, out of however many births, this amount of people could be walking around or be born with a CACNA1A mutation is 1 out of 11,700, or it's easier to think of as at 8 out of every 100,000 people are living with a CACNA1A mutation. Whether or not it means that it's pathogenic and they have the disease, that still, needs to be teased out. And then this is a word cloud that, I generated just based on what types of symptoms there are and which ones are more prominent. One thing about this disease is that it is very heterogeneous in terms of symptom presentations. And then there's a lot of variance in how severe these symptoms can present. So a lot of our kids have that ataxia, developmental delay, problems with communication. They have intellectual disability. Some of them have seizures, eye movement disorders, and so on. And we can actually even quantify that too. This is some data from our natural history study, which collects information from families over time. And so you can see, and this is from a cohort of about 100 individuals, that movement abnormalities like ataxia, movement disorders, are reported in nearly 80% of the patient cohort. Clumsy, being uncoordinated, the hypotonia again, abnormal brain morphology. I don't have EEGs on here, but a lot of our kids also have abnormal EEGs. And then if we look into like development, you can see again, a high percentage of these individuals have delays in gross motor skills, fine motor skills, language delay, speech impairments, and then a portion of them also do show some developmental regression. And our community does continue to grow. Again, we don't really have an accurate count of how many people are living with this disorder, but one thing that we have done as a foundation is to create this contact registry where we have a website, we have events for families around the world, and we just let them reach out to us and tell us, who they are, where they live, so we can start to see where everybody is. And so you can see back in 2020, when the foundation started, we were engaged with about 60 patients, and you can see there's this nice growth over the last 5 years. And at this moment, we are, engaging with over 500 patients. This is worldwide. This is a list of all the different countries in which some of these individuals live. And I would say, I think over 50% of this total population is inside the US so far, but we do try to serve a global community. And so this also tells us that there is what we call this high unmet need, right? There's this a large patient population out there that have really no good treatment options, no good care options. And so that's something that we can use to try to, negotiate with pharma companies, right? Saying, look, if you want to make a difference, you can invest in our community. And knowing how many patients there are and knowing where they are physically in terms of, clinical trials, that's really helpful because, clinical trial sites could be where there's a large population of the patients, and that would, be a good thing for increasing enrollment in clinical trials. And it's ultimately better for sort of everyone who's invested. So these are some of the initiatives that we are involved in. I'll go into just a little bit of background in terms of what CACNA1A is. So the CACNA1A or CACNA1A gene stands for the calcium voltage gated channel subunit alpha 1A. which is a really long word, right? Which is why we call it CACNA1A. But this is a gene that's important for neurotransmission or this communication within between neurons. And I think we all understand how important that is. So, if we think about the brain and we kind of like focus in on that micro level of neurons. And these are those specialized cells that basically function normally, that allow us to walk and talk and eat and learn and do everything that we can do. And that communication within the brain and even within the brain to the rest of the body really relies on these electrical signals. So that's the form of information in the brain. It's in the form of these electrical signals. And so if you think back to neurobiology, these neurons form this big network, and a neuron gets a signal from a neighboring neuron, and that basically allows for this electrical signal or an action potential to propagate down the neuron, and then it sends a message to the next neuron, and then it propagates that action potential, and so on and so on. And you can imagine that within the 86 billion neurons that we have in our brain, right, that this is a very tightly regulated process. Things have to happen, at the right time in order for us to be, functioning human beings. But one important thing here is that this process of neurotransmission does rely on ion channels. And so these are proteins that are found in these neurons. They make a little hole or a little pore in the membrane. It allows for the exchange of ions like sodium, potassium, and calcium across the neuron membrane. And that helps to propagate that electric signal down the neurons and then again to the rest of the body. And so CACNA1A, that gene encodes for a specific type of calcium channel. CACNA1A is globally expressed in the brain, but it's really enriched in the cerebellum. So the calcium channel that is encoded is called CAV2.1. This is one of 10 calcium channels that are found in our body, but this one, again, is highly seen within the brain. And it's voltage gated because it relies on, again, the changes in the electrical signals across the membranes of these neurons in order to activate, to open, and also to close. So this is a schematic of the secondary structure of what these ion channels look like. So we have these 4 sort of pseudo-homologous domains. They're not completely identical, but we have these 4 domains, and within each of these domains, we have these 6 transmembrane or membrane-spanning subunits. And there are the S4 subunit in each of these domains is called the voltage sensing domain. And so it has the job of activating the entire channel once it senses those voltage changes across the membrane. And then S5 and S6 of each domain are the little subunits that physically form that pore. It like lines that hole in the membrane. And so I don't have a good 3D structure of this because we don't really have a good crystal structure of that pore domain yet. But you can imagine, if you can, that these 4 domains sort of curl up on each other in such a way that all of the S5 and S6 domains are, again, forming the pore, and then the rest of the subunits are on the outside of it. So CAV2.1 drives this neurotransmission at the presynaptic membrane. So the synapse is, again, that space between neurons, and that's where a lot of this information is transmitted. So if we take these neurons and we kind of blow it up, this is my very crude drawings of what is happening, between these neurons. But we have a presynaptic neuron, we have a postsynaptic neuron. And so CAV2.1, this calcium channel, is at the ends of the neurons, or these boutons. And what happens is that when this action potential reaches the end of the neuron, these calcium channels open and allows calcium ions to come in. And that's then signals for these neurotransmitters, right? These chemical messengers in our brain to be packaged up into these little vesicles. And then those vesicles travel across the synapse. They get to the postsynaptic neuron, which many times then induces some sort of action. So either that action potential says, please continue this information, or in some cases in inhibitory neurons where it actually might stop the activity. Because you can imagine at some points you might want to dampen or turn off activity at some points, and then you want to turn it on again. So when you think about how this protein and how these mutations in the gene can lead to these pathogenic or disease-causing symptoms, we kind of we kind of crudely separate out these mutations into two different categories. And it's not really black and white, but in some ways we can think about how these mutations in the gene may lead to something called a loss of function mutation or variant. And this is where we have less calcium entering cells, and that could be due to different types of disease mechanisms. So when we talk about this P naught here, this is the probability of opening. And so in some cases, this probability of opening can be reduced. So this channel somehow opens later, or it's harder to open. Another reason why there could be less calcium entering cells is that there's a decreased level of the functional protein. This could be due to trafficking defects, because if you think about a neuron, the cell body is at one end the cell body is one end of that cell. That's where the nucleus is. So that's where everything's made. And then all these proteins have to get down the axon, right, to where it needs to be. So there could be, one of these mutations could be disrupting that process of trafficking these proteins to where they need to be, or they could be causing these proteins to be unstable and then not, expressed where they need to be at all. And so you can think of that as like less information being sent. And then on the other hand, we have these gain of function mutations or variants. So we have more calcium entering cells, and that probability of opening is actually increased. So maybe this is a channel that opens earlier, or it's easier to open. And you can think about this as your brain sort of being overloaded by information. And so at the foundation, one of the things that we want to do is to advance this research and to help our research community learn or think about how we can treat these disorders. So again, if we think of it in this really simplified concept here, if we're dealing with these loss of function variants, you can imagine that we want to find a drug, right, a medication or a molecular tool that can open the channel, increase the activity, to stabilize the channel, or to help traffic that channel to the ends of the neurons where it's needed. And then for those gain of function variants where there's too much activity, we want to find a drug or molecular tool to close that channel, to destabilize it, or again, prevent it from going to the ends of the neurons. And so these are, again, some of the general approaches that we think about when we're trying to develop treatments. And so, we can also think about it from a genetic point of view. So the CACNA1A related disorders is an autosomal dominant disorder. So it's located on one of the numbered chromosomes and not one of the sex chromosomes. And you only need one copy, right? Because remember, we all have two copies of each gene. In this case, you just need one copy of that mutation in order to cause or to lead to the disease. And so the idea of these treatments is that we want to restore that normal balance of calcium influx. So ideally, we'd love to be able to get rid of that mutated allele and replace it with like a healthy copy of the gene. Again, normal channel restored, neurotransmission resumes. But there's a really big problem in terms of the technology that we have, and that is that targeting the allele that has that specific mutation is still almost, I wouldn't say impossible to do, but very difficult to do. A lot of the approaches that have been developed, you can, you're sort of targeting both. And so you can imagine that there might be some cases where if this allele is, or if that copy of the gene is degraded because maybe it's a truncation mutation, so nonsense mediated decay might get rid of that gene copy, then it doesn't matter, right? Because you might still be able to upregulate the wild type copy. But in cases where this is a very harmful allele, you don't want to be trying to upregulate that either. So we're still trying to think through how to get things, into the brain and replace or target that mutated allele and get that healthy copy in. So I'll go through this really quick. So how do we get to these specific treatments? This is a really just, again, simple schematic of how we start with the rare disease population with the intent of getting to clinical trials. And so when I talk about basic research, this is the research that I think many of us here are trained to do in terms of the fact that we're like using the systematic approach to answer a question. So again, thinking about disease mechanisms. What happens if you knock out this gene? What happens if you put a mutation in this of this part of the gene, right? Just answering, again, like a really straight, here's my hypothesis, let's test it. And then translational research is something that I think is kind of coming into academia a little more, but still not as much as maybe it could. But this is taking those basic principles from basic research and then applying them to humans and specifically into patients. And so once, you start to understand what's happening in the disease, you can start to think about what to develop, and then get to what we call these proof of concept studies. And so these are experiments that are usually done in disease models that show that this disease is amenable to that treatment. And that's really important for the FDA to see. Eventually, what we'd like to do is file an IND application or an investigational new drug application. If that's approved, then we can put that treatment into a clinical trial. And then again, hopefully get to a point where this new medication can be approved for our community. I'm going to skip this. This is basically our research roadmap. It's just another way to show what I was just showing before. And so one of the things that, again, we've been doing at the foundation to advance research is that we have launched all these different initiatives to get our community ready for clinical trials. We do offer a grant program to support research. Again, we promote collaboration, and we're building what we call these preclinical assets. So either patient-derived cells as a disease model or animal models too. And something that I really want to emphasize here too is that when it comes to these rare diseases, patient data is really what drives research because you don't know a lot about these diseases. So you kind of have to go back to that source and figure out what is it like for people who are living with this disease. So we have a couple different data collection programs that allow us to collect data over time. So we understand that evolution of the disease, which tells us what types of treatments might be needed. And we have both caregiver and patient-reported data. We also have clinician-reported data. So what are the doctors saying? That gives us the most comprehensive understanding of the disease. We also ask what's important to the community, because you can imagine if scientists are working on a treatment that isn't really relevant, there's kind of no point because the patients aren't going to be interested. They're not going to enroll in a clinical trial, and that's like a big failure there. So when we ask our community what they want, you can see in terms of the top symptoms that impact daily life the most, ataxia, right? That movement disorder is very high, trying to improve communication, trying to reduce seizures. When we ask about the daily activities that are a struggle, right? Being independent, communicating, performing self-care activities, those all require a lot of help from the caregivers there. And then when we ask them, what are the goals for future treatments? Again, you can see decreasing ataxia symptoms, decreasing seizures, improving communication. And this is really important too, slowing or stopping the progression of the disease. A lot of the medications that are currently being used only treat symptoms, right? So they might reduce the seizure, they might help you walk a little bit better, but it's not actually addressing the root cause of the problem. So it's kind of like putting a Band-Aid over a wound that is going to open up at some point. And so what we can do is basically take that patient data and start to build these research tools. So we do have a biorepository where we have generated patient-derived iPSCs, and iPSCs are these induced pluripotent stem cells that basically are stem cells that can be reprogrammed from blood cells, and then those iPSCs can be programmed into any type of neuron. So you can imagine that these are pictures of some of these cortical neurons that have been grown on plates in a lab. And these are all the different mutations for which we have a patient-derived line. And some of these patients, again, are gain of function. Some of them are loss of function. Some of them are very severe. Some of them are a little milder. So with that, again, we have this comprehensive box of tools that researchers can use. We also have a lot of different animal models, both in mice, fruit flies, and worms. And so again, lots of different disease models that can be utilized for research. Our grant program, we do offer these $50,000 seed grants to support both basic and translational research. These are some of the institutions that we have granted funding for. So we're really proud to be able to have been able to support really important research in terms of understanding the disease mechanisms, right? How does a mutation impact the protein structure and function? How does that lead to the symptoms? Again, these new therapeutic approaches, we're also funding things like that. Again, promoting collaboration. We have a global research network where we have sort of forced everyone to come to these in-person research roundtables and force them to think about our disease. And it's been pretty successful the last couple of years. Last fall, we weren't able to hold an in-person roundtable, but we were able to hold a virtual one. So this was a nice screenshot of everybody. Again, these are families, researchers, people from companies, people from NIH, all coming together to help us think through. I'm going to skip over this again. This is just saying how do we treat CACNA1A related disorders. So again, with the research that we've been supporting, this is what we call our therapeutic pipeline, where we have things like a gene therapy that's in the works. We have something that can target protein stabilization and destabilization. We have some RNA therapeutics with ASOs, or these antisense oligonucleotides that can physically bind to the gene and can either turn the gene on or express it at a higher level to increase activity, or it can do the opposite, where it can knock down gene expression and then decrease activity. We're also doing drug screens where we can take medications that have already been approved by the FDA and test them on our models. And sometimes they might actually work for our symptoms, so we can kind of do that as well. So this is sort of a combination of everything that we have been supporting in the last couple of years. I'm going to get rid of that slide. But a really important thing and a really important milestone for us as a foundation is that we just had our very first clinical trial for CACNA1A related disorders announced. This is working with IntraBio. We're finally in someone's pipeline, which is really exciting. And this is a small molecule that's supposed to help with neural inflammation and ATP production. And so there's a chance that it could really help improve some of the symptoms for our community, and they will start recruiting in the fall of this year. And then I also wanted to say that every year we do have a really big awareness day. It's always February 28th, or in a leap year, it is the 29th. So tomorrow is Rare Disease Day, and there's a lot of different institutions that hold Rare Disease Day events. There's like buildings around the world that light up for Rare Disease Day. It's also a big fundraising day for a lot of these patient advocacy groups. So I'll end there, and thank you very much for listening to this. I think I talked a little longer than I intended to, but I'm happy to take questions.
Thank you so much, Pangkong.
Yeah.
I'm going to let you drive and see if there are any questions.
Sure. I mean, again, that was a lot to take in, but I'm happy to talk through maybe some of the specific like therapeutic approaches we have, or happy to talk a little bit more about how this disease has impacted our family. Anything's really on the table. But I think what I also want to say is that this is, one of those non-traditional pathways for a PhD. And, in the nonprofit sector, it's not about making a lot of money, but it is about making impacts. So if you are passionate about a rare disease, or you want to help, a community out there that's in need, there are certainly opportunities to share your skills and your talents with the Rare Disease Organization. Okay, I have somebody who says, yes, are you doing treatments for Alex? We are at the moment. We were actually able to access one of the drugs off-label, meaning that it's not approved for CACNA1A related disorders, but because his disease is similar. We were able to get a hold of that. And so it just treats symptoms. It's supposed to treat ataxia, but we're actually seeing some improvement. So he's been on this medication since August, and we have seen his speech explode. He just talks so much more. He's got new vocabulary. He says things that like I didn't even know he knew what that word was. Um, and yeah, I think his overall movement is just better. He got back on his bike. We have an adaptive bike for him. He hasn't ridden it in like 2.5 years. He got right on and he started riding again. So that is something where, yeah, I mean, that's like one of the advantages of being in an organization like this and being able to access things off label. And this is something that we also encourage our families to do. In terms of things like a gene therapy or something that's a little more invasive. I think for me, like if somebody said tomorrow we have a gene therapy ready for Alex, I personally would probably say no because he's on the milder side of the spectrum. But we have some kids who are in and out of the hospital because their seizures are so horrible, or they have these hemiplegic migraines that look like a stroke, but they're neurological in origin, and that can lead to, brain swelling, coma, and even death. Um, so for me, I would say no because things like this new medication have stabilized Alex. And a lot of the gene therapies right now are very invasive, where you actually have to sometimes drill a hole in the brain and inject this vector that's carrying a new copy of the gene or carrying this new molecular tool to get it directly into the brain. And that can have some side effects. As you can imagine, the physical side effects, and then also, a lot of these genes are being delivered using a viral vector. Even though like the parts of the virus, the viral vector that are pathogenic are taken out, it can still induce some sort of like toxicity. So your brain might still recognize it as a virus, and you could have like a really severe adverse reaction. So for me, I think I have the time to wait until maybe these new techniques are a little more, I don't want to say safer, but that they're a little more efficient and they've been used a little bit more. But we definitely have families in our community that would hop on that, would take a gene therapy the next day.
I have a quick question related to that.
Mhm.
I've been hearing about every cure, and is
Yeah.
that sort of how you figure out what medications might work for you? And
Yes, so that is one, yeah, that is one way, one method. I've met David Fagenbaum before, who leads Every Cures. And again, he's just got one of these great stories where he sort of found his own sort of cure for his disease. And not all of us have those great talents, but he has started this company where you can use AI. So you can use AI to scour published literature, I think to scour even like drug libraries for different companies, and you can kind of see which ones might be able to treat your disease based on the mechanism of action and things like that. And then depending on whether that's a drug that's readily available already, or if it's something that has to be manufactured, those can sometimes be used to treat, again, the symptoms of patients while they're waiting for something like a gene therapy. So we haven't worked directly with Every Cures, but when we do drug repurposing with some of the scientists, it's sort of the same idea, except that it's actually doing the physical testing in cells versus AI. But certainly we could do a combination, right? Use AI to find candidates and then test those candidates in the cells. So yeah, there's a lot of different approaches, and everything costs money. So that's another thing too, is like we're doing all grassroots fundraising right now. Um, so even those $50,000 seed grants are really hard for us to raise, but and as scientists, that doesn't go very far in terms of research. So we really try to use those grants as leverage so that these researchers can sort of generate some preliminary data that could be used to get like a larger R01 grant from the NIH.
Yeah, maybe I just want to make a couple comments too,
Yeah.
Pangkong. Thank you so much for the talk. I think it was really informative. And I think having a kids is hard. Having a kids with some of these rare disease, I can imagine how challenging that must be for a parent. And so, I really commend you for doing all of this to try to spread the word and to inform the public more about it. I mean, a lot of the things that we do, even science, our apparent or, foundation, it's really hard to find the cure. But I think the work that we do, like you said, does make an impact long term,
Absolutely.
like eventually year after year, if we keep doing research, that eventually somebody might find something that will be really helpful to people, right? And I think that's really the goal.
Mhm.
Like maybe it's not to cure, but just really to improve the
Yeah.
lives of those who are infected so that they can live normal life. And so I think that's really great what you're doing, and I appreciate all of that work. A question I have is, for kids who have it, whether they're milder or maybe more severe form, does the condition get better as the person age over time into adults, or does it get worse? What is the typically, symptoms for a person with this?
Sure. Yeah, that's a really good question. So to any of the current medications that we are using. And so there's sort of like a control for us, right? Like if you're not treated, this is what the trajectory looks like. But unfortunately, we don't know what it really looks like for, every single child. Um, But I would say anecdotally, when we talk to parents, whether or not your child is very, very severely impacted or mild, we do see that as these kids turn into young adults and into, older adults, that there is this regression in skills. And I think a lot of it is due to the fact that, when you think about like my son is in, he's in fourth grade and he has inclusive classrooms at his school. So he's in with his peers as much as possible. And then he gets pulled out for like one-on-one, academic teaching, but he's constantly stimulated by the environment around him. And he's going to have that up until high school. And then depending on what he wants to do, right? When he turns 18, he's legally an adult. But what happens is that his care, right, as an adult, every support system that was built in for kids it's very different now. There's not a lot of support systems built in for adults to begin with, much less adults with disabilities or diseases. So I think, some of these adults that we know, they get depressed, right? There's implications in like psychiatric illnesses because they lose that stimulation. They lose those connections. And I think that really does have a really big impact on that. So that's what we know right now. But we're again hoping that with the advances that are being made in terms of technology and treatments that we might be able to treat people, sort of as young as possible so they can have this better outcome for their life. So again, really appreciate this opportunity. And I think one thing that I like thought about doing, but I have no bandwidth, is again trying to raise awareness within the Hmong community. And I think not even rare diseases, just any diseases or any disabilities. I'd like to do that one day. I don't know if there's anyone within this organization that is sort of in that space, but I do feel like I'd love to work towards just more awareness within our own communities.
I think that's the goal that hopefully, part of what we're growing as an organization is that,
Mhm.
that's something we will do. And I definitely share your frustration with that. It's not only just rare
Mhm.
disease in terms of like alcohol drinking, in terms of
Mhm.
cancer, in terms of a lot of these, conditions that most people have, really just to
Yeah.
convince them, to really do a certain act a certain way or to really study it to understand more. Some of these are become more like taboo topics and not people
Yeah.
talk too much about it, right? And it's just things that happen, like especially rare diseases, this spontaneous mutation that happens in
Mhm.
somebody, and understanding ways to improve that would be great, and allowing Hmong people to find the right kind of help to help their
Yeah.
kids, right? You are a scientist, and so at least you know what's going on, you understand all these mechanisms, and you know how to get your kid, the support that they need, so that even if this thing may not make their neurons as active, they're getting all the stimulation around by their teachers, the environment, and that part plays such a huge role in their brain development and function as well, right?
Yeah.
But if you're a Hmong person, like a normal average person who has a kid like this, they may even just put this kid away, and hide or something like that. And that the damage that done over time is this kid not being exposed and their brains not being as stimulated as it should be, so that they will have, hopefully, will make their condition not as severe as it should be. And so, there's little things like that that we need to teach, that we need to try to reach out to the Hmong community so that they're aware of that. And so definitely do that. And I think if there's any opportunities that we can help you, or if we all could contribute to try and do that some more, I think we should do that to really get Hmong people to think more of that. It's not really a cure. Like Hmong people, like we're sick, we want
Yeah.
to cure,
Yeah.
and it doesn't have to be to cure us or our immediate
Mhm.
person, but it's to make it better for other people in the future, so that they don't experience the same kind of frustration, that's the same kind of burden that you do. And so I think that sort of thing, it needs to go into the Hmong community more and to discuss more. But I think it's also just education, right? And I think
Mhm.
maybe our generation going forward will be more open-minded and more willing to engage, and also, make sure that the community do what it needs to. But we definitely have to do something to bring
Yeah.
that awareness into the community.
Mhm. Yeah, well said. Happy to help out.
Yeah, I think the conversational science piece that you talked about, Ping Pong, earlier is so important, because before we can even talk about some of these, challenges or health concerns or diseases, you have to spend a lot of time just explaining genetics, right? And that's really difficult for any lay community, but even more so for our community, and finding the right words to explain it all. And so I think that's really important. I wonder if there's a way we can think about, as HND continues to do their conferences, maybe, because we were just at the one last year. I'm sure they'll have one in a few years. That would be really cool to have more of a biomedical, more of a health focused session. That's also more, kind of educating folks on these things, and then with a science slant, because there is a science slant to it for
Yeah.
sure. So,
Mhm.
yeah.
Yeah, this actually reminds me of a conversation that I had with Du Yi's brother when we were at H&D, because your brother is a physician, right? And I was remember talking to him about like Hmong people were had a predisposition to have diabetes, but we also eat a lot of rice. And I was like, how should we tell our family that like maybe we should cut back on the rice? But it's
Mhm.
tough because it's like a staple food for us, you know?
Mhm.
So I think these conversations are important to have. Like these habits are hundreds, or if not thousands of years old. But I
Mhm.
think yeah, there's a lot of work to be done. I think it'd be interesting to have some sort of like community health stance if we go back to H&D, like community health, but also science would be awesome.
Yeah.
And I really enjoyed all the work that you've done for the foundation. I was really impressed by, the establishment of model cell lines and model animal models to study this, because I think there's so much work that goes into doing that and like QCing those types of models. And the place where I worked, the first 10 years of the organization was just focused on making cell lines. That was just all they
Yeah.
did. They're like, let's just make cell lines for the community. So I'm interested to see like where I work maybe in the next 10 years. I wonder if there's any way that we could, go back and try to make, disease cell lines for rare
Yeah.
disease and disseminate those. But no, I
Mhm.
really appreciate your talk. Thank you.
Yeah, you're welcome. Yeah, no, we're really fortunate that there are, again, institutions out there that we can collaborate with to generate these lines and to generate the models. We work really closely with Jackson Laboratories. It's another nonprofit, but I think they also get a lot of funding from NIH, and they have this rare disease translational research center. And so a lot of times we're able to get mouse models generated through them. And then again, also really lucky that we have this research network that can characterize those models, because again, everything everything costs money. So sort of like if we can make the models and then scientists can take that and do the work, then it's been a good investment. Okay. I think
All
we're at
right. Well,
time, right?
yeah, we're at time. Thank you so much, Pangkong. This
Mhm.
was so insightful and really appreciate all that you do.
Mhm.
Also interesting to hear it from a different slant, in terms of
Yeah.
how our research and thinking forward and how we move forward in our careers and how it can have
Mhm.
lead to real impact. And so I hope we stay engaged and hope to see
Yeah.
you future meeting.